Yeah, man. Good morning. Hello.
Hello, good morning, sir.
Good morning. How are you, man?
I'm good, sir. How are you?
Fine, man.
Mm.
Shall we start, man?
Yes, sir. We'll start.
Yeah, please.
Okay. So, hello everyone. Welcome to Tawhid Foundation at Allied Healthcare Sciences, Bangalore. Today's class focuses on biomedical waste management, a vital area in healthcare. So, I'll give a brief introduction about what biomedical waste management is and why is it important to study. So biomedical waste includes any waste product during medical treatment, diagnosis, research or related healthcare activities. This waste is unique because it can carry infectious agents, toxic chemicals or sharp materials, placing healthcare workers, patients and the environment at risk if not handled properly. So, effective biomedical waste management is essential to prevent the spread of disease, protect the environment from contamination, and ensure the safety and compliance of healthcare facilities. By understanding how to properly segregate, treat, and dispose of biomedical waste, we help safeguard public health and uphold professional standards. So there are five objectives of biomedical waste management. First is to define biomedical waste and its sources. Second, to classify biomedical waste based on risk and type. Third is to explain collection and segregation step. Fourth is to discuss treatment and disposable method. And last, fifth is to highlight health and environmental risk. I'll explain each objectives. First, to define biomedical waste and its sources. Here, why it is important, because to know what is biomedical waste and its sources of generation, to gain knowledge about the different types of biomedical waste, to explain the steps of biomedical waste management, and lastly, to describe the proper mode of collecting and segregating the biomedical waste. Second, this first objective mainly aims to help you understand the definition of biomedical waste generated as a result of medical treatment, diagnosis, research, or immunization in healthcare settings. Sources include hospitals, clinics, laboratories, blood banks, veterinary centers, and even home healthcare centers. Second, to classify biomedical waste based on risk and type. So here you will learn to identify and classify biomedical waste based on its characteristics and associated risk. Type includes infectious waste, pathological waste, sharp needle, syringe, chemical waste, pharmaceutical waste, and general non-hazardous waste. Each type requires specific handling and disposable method. Third, to explain collection and segregation step. This objective focuses on the systematic approach needed for safe handling of biomedical waste. It includes segregation at the point of generation, proper collection in designated containers, safe transportation, storage, treatment, and final disposable. Fourth, to discuss treatment and disposable method. Here, proper correction and segregation means using color-coded bins or containers to separate different categories of waste right where it is generated. This reduces the chance of cross contamination and makes further treatment and disposal safer and more efficient. Fifth, to highlight health and environmental risk. This objective is about recognizing the health risks such as infection, injury, or exposure to toxins and environmental hazards caused by poor biomedical waste management. It also covers standards treatment method like incineration, autoclaving, chemical disinfection, and secure landfilling, as well as best practices for safe final disposal. These objectives will help to gain the knowledge and skills required to manage biomedical waste responsibly, minimizing health risk and ensuring regulatory compliance. So, what is exactly biomedical waste? The definition of biomedical waste is biomedical waste refers to any solid or liquid waste generated during the diagnosis, treatment, or vaccination of humans or animals. This include a wide range of materials such as bandages, syringes, body fluid, laboratory cultures, tissues, discarded medicines, and chemical waste from healthcare facilities. So, the significance of biomedical waste lies in the hazards primarily due to two main reasons. First is infectivity, and second is toxicity. So, infectivity, biomedical waste often contains pathogens, bacteria, viruses, fungi, or parasites that can cause infections and diseases. For example, blood soak material, contaminated sharp materials, and pathological waste carry a high risk of transmitting infections like diseases, hepatitis, tuberculosis, HIV, and other contagious diseases. When these wastes are improperly handled or disposed, they can infect healthcare workers, patients, waste handlers, and even the general public. Second is toxicity. So, besides infectious agent, biomedical waste may contain harmful chemicals, pharmaceuticals, or radioactive materials. Chemical waste include disinfectants, laboratory reagents, and expired medicines that can poison humans, animals, and damage the environment. Toxic substances may contaminate soil, water, and air, leading to a long-term health care such as chemical burns, poisoning, or cancer. So, because of this dual infectivity and toxicity, this dual hazard, infective and toxical biomedical waste demands careful management through segregation, treatment, and safe disposable methods to protect human health and the environment. So, proper handling prevents the spread of diseases and minimizes environmental contamination, ensuring healthcare settings remain safe for all. This figure represents biomedical waste production in hospitals. So this diagram represents the biomedical waste production in hospitals, specifically focusing on data from AIMS Delhi in 2018. This diagram is representation of AIMS in 2018. So it highlights the different categories of biomedical waste generated by hospitals and the amount produced monthly. So I'll give breakdown of these biomedical waste products. First is yellow waste, which is 24,000 kilograms. This category typically includes infectious waste such as contaminated items like dressing, cotton swab, human tissues, and body fluids. Yellow bags or containers are used to segregate these type of waste because it possesses a high risk of infection and requires special treatment such as incineration or autoclaving. Second is plastic and rubber waste, which is 23,500 kilograms. This includes disposable medical plastics such as syringe, catheter tubes, gloves, and other rubber-based items commonly used in healthcare. These materials require segregation because they are often non-biodegradable and need appropriate disposal or recycling methods. Third is sharp waste, which is 500 kilograms. So sharp waste includes needles, scalpels, blades, and other pointed instruments. Though it is the smallest quantity compared to other categories, it poses a significant risk of injury and infection. Sharp waste requires safe collection in puncture-proof containers and sterilization before disposal. Last is glass waste, which is 26,200 kilograms. Glass waste include vials, ampoules, slides, and broken glassware used in laboratories or medical treatment. Glass waste is hazardous due to the risk of cuts and potential contamination. It must be handled carefully and often requires special disposal methods like sterilization or encapsulation. So this overall significance, AIMS Delhi produced approximately 74,500 kilograms of biomedical waste per month, around 2,500 kilograms daily. So reflecting the enormous volume of waste hospital generate. Understanding these types and quantities of waste helps in planning and implementing effective waste management protocols, ensuring proper segregation, treatment, and disposal. So each category of this waste requires distinct handling methods, emphasizing the importance of biomedical waste segregation at the source to minimize health and environmental risk. So, this data underlines the critical need for strict biomedical waste management practices in large healthcare institutions to prevent contamination, protect healthcare workers, patients, and the environment. Sources of biomedical waste. So there are two sources of biomedical waste, which first is major sources, second is minor sources. Major sources include government and private hospitals, nursing homes, dispensaries. Second is minor sources, which include physicians clinic, dental clinic, labs. So, the source of biomedical waste is the place or the location at which biomedical waste has been generated. The source of biomedical waste, as I mentioned, is classified into two, major and minor. Major source generate more amount of biomedical waste compared to minor source, and also there is regular generation of biomedical waste in the major source, which includes all this government hospital, nursing homes, and dispensaries. And minor source includes these physicians and dental clinics. Sources of biomedical waste. So these are the primary generators of biomedical waste, producing large quantities due to the scale and nature of their healthcare or research activities. This diagram illustrates the sources of biomedical waste divided into two categories, major source and minor source. So in hospital, the biggest contributors generating waste from various departments like surgeries, patient care, laboratories, and emergency rooms. Labs, it includes diagnostic and research laboratories, produce waste from testing samples, cultures, chemicals, and reagents. Research center, institution focused on medical or biological research generate waste related to experiments and animal studies. Animal research facilities conducting veterinary or experimental research with animals produce biomedical waste, including tissues and bedding. Blood banks, these handle blood donation and storage, generating waste such as used blood bags, needles, and testing materials. Nursing home long term care facilities generate waste from daily patient care, including dressing and disposable. Mortuaries, places where deceased bodies are prepared for burial, generating pathological and infectious waste. Autopsy center facilities performing postmortem examinations produce significant amount of pathological and infectious waste. Minor sources, these sources generate smaller amount of biomedical waste but are still important contributors and require proper waste management. Clinics, smaller healthcare units that treat outpatients generating medical waste such as used syringe and dressing. The second is dental clinics generate specific waste like extracted teeth, used dental material, and sharp materials. Home care. Home care include waste produced by healthcare activities in patients' home like used needles or bandages. Cosmetic clinics. These generate waste from minor surgical and cosmetic procedures. Paramedics, first responders, and emergency medical services produce sharp and disposable medical materials. Funeral services generate waste during preparation and handling of bodies. Last is institutions. Other facilities such as nursing college or veterinary institutions may also produce biomedical waste. So, understanding the diverse sources of biomedical waste is crucial for targeted waste management strategies. Major sources handles large volume and often require specialized disposal system, while minor sources need inclusions in community wide biomedical waste programs to ensure safety and compliance. So proper identification of these sources enables effective segregation, collection, and treatment, which overall reduces health hazards and environmental impact. Classification. Classification of the biomedical waste is carried out based on its characteristic source of generation and the level of hazard to the environment. The biomedical waste is classified into two types, non hazardous waste and hazardous waste. So, non-hazardous waste, about 75 to 90% of biomedical waste characteristics were similar to that of domestic waste and are non-risky in nature. This waste is generated mainly from the organization and maintenance of hospital and healthcare centers. Hazardous waste makes up about 10 to 25% of the total biomedical waste produced in healthcare settings. These wastes are hazardous because they can cause infections, injuries, or environmental pollution if not managed properly. So, infectious waste. This includes any item contaminated with blood, body fluids, or pathogens like bacteria or viruses. Examples: soil dressing, bandages, swabs, and items exposed to infected patients. These can transmit disease if handled improperly. Second is pathological waste, consists of tissues, organs, body parts, human fetus, or body fluids like blood removed during surgery or autopsy. This waste can be infectious and require careful disposal. Pharmaceutic waste, unused, expired, or contaminated drugs and vaccines. This includes items like antibiotic, cytotoxic drugs used in cancer therapy, and materials that have come in contact with them. Some of these chemicals can be highly toxic. Chemical waste includes discarded chemicals from diagnostic work, cleaning, disinfectants, solvents, reagents, and laboratory chemicals. Many can be corrosive, toxic, reactive, or flammable. For example, mercury from thermometers, formalin, and solvent waste. Radioactive waste contain radioactive substances like those used in radiation therapy or diagnostic imaging in hospitals. This waste can come from unused radiotherapy liquids, lab research, or medical tests using radioactive materials. Sharp materials, items such as needles, syringe, scalpels, blades, and broken glasses. All sharp materials can puncture or cut skin and are often contaminated with infectious material, making them double hazardous. Last is heavy metals. It includes things like batteries or medical devices containing lead, cadmium, or mercury. These metals can pollute soil and water if not disposed correctly and pose health risks to human and animals. So all these categories must be carefully managed to prevent harm to healthcare workers and public. Classification of healthcare waste. These flowchart illustrates these diagram classification of healthcare waste, dividing them into major categories based on their potential risk. So, healthcare waste are broadly classified into two main types: hazardous waste, which is 10 to 20%, non hazardous waste, which is 75 to 90%. These wastes pose significant health or environmental risk due to their infectious, toxic, or physical properties within hazardous waste. So, infectious waste, which is 15 to 18%, contains pathogens that can cause infections in humans or animals. This includes waste contaminated with blood, body fluid, cultures, or swabs from infected patients. Additional hazardous waste types include sharps. Again, this include needles, scalpels, blades, pointed or sharp objects that pose injury and infection risk. Non-sharp materials, other potentially infectious waste that is not sharp, such as contaminated dressing or gloves, plastic disposable, single-use plastic healthcare items like syringe and tubing contaminated with infectious material. Liquid waste includes blood, body fluids, laboratory cultures, or chemicals in liquid form that could be infectious or toxic. Non-hazardous waste 75 to 95%, 90%. So these represent the majority of healthcare waste considered less risky but still requiring proper disposal. This category includes radioactive waste, Materials contaminated with radioactive substances used in diagnosis or treatment, example radiotherapy. Second is chemical waste. It includes disinfectants, solvents, and reagent that can be toxic or corrosive. Cytotoxic waste containing substances toxic to cells such as chemotherapy, drugs, or their residues. Pressurized containers, gas cylinders, or containers under pressure which pose explosion or hazard risks if mishandled. So these classification helps healthcare facilities prioritize handling, treatment, and disposal method depending on the risk levels of different waste. Hazardous waste requires strict segregation, careful handling, and special treatment to prevent infection, injury, or toxicity, while non-hazardous waste must be managed to prevent environmental conditions, contaminations, and ensures workplace safety. So proper categorization is essential for effective biomedical waste management. Hazardous waste in examples. So I already explained this hazardous waste example in this classification. I'll just explain the terms. Infectious, which include culture, swabs, excreta; pathological, tissues, body, blood parts; pharmaceuticals, expired and contaminated drugs; chemicals, reagents, and disinfectants; radioactive therapy waste; sharp includes materials like needles, blades, and scalpels. Biomedical waste management, why it is needed, and key steps. So biomedical waste management, proper management is highly essential since it induces various risks to the human health and to the surrounding ecosystem that leads to the ecological hazard, professional hazard, and public hazard. So the healthcare waste management process involves several detailed systematic steps to ensure safe handling, treatment, and disposal of biomedical waste, protecting health workers, patients, the community, and the environment. So, a detailed explanation of the important stages in the process I'll give. First is segregation. So, segregation waste must be separated using desired color coded bins or containers specific to waste types. Example yellow for infectious waste, red for plastic waste, and blue for glass. Then collection. Waste should be collected in secure, leak-proof bags or containers and sealed properly. Storage, before storage, one step is there which is pre-treatment. So certain waste, example infectious laboratory or highly infectious waste, require pre-treatment at the healthcare facility before transportation. For example, autoclave. Then storage, temporary storage areas within healthcare facilities should be secure, clean, and again leak-proof with restricted access. Storage duration must be limited, generally not more than 48 hours, to prevent infections and odor problems. Transport, waste is transported safely within the healthcare facility or treatment areas. Treatment method depends on waste type, incineration for pathological waste, autoclaving for infectious waste, chemical treatment for liquid waste, and shredding for sharp object. So the goal is to render the waste non-infectious and safe before disposal. Last is disposal. Treated waste is disposed of according to regulation, secure landfill for non-hazardous waste, encapsulation for sharps or specialized disposal for radioactive waste. So disposal sites must be environmentally secure to prevent contamination. Segregation. To avoid mixing of the biochemical waste with other, a container should be set to the side with color coding bags at the point of generation. The sorting or separation of waste into different categories is referred as segregation. Segregation will decrease or minimize the risk in addition to rate of managing and disposal. Segregation is the most important and critical steps in biomedical waste management. Only effective segregation can confirm the effective biomedical waste management. So, how does it help? Using color coding systems. These color codes represent the segregation system for biomedical waste, which is critical for safely managing different types of healthcare waste. Each color corresponds to a specific category of waste based on its characteristics and the treatment method it requires. So, yellow color bin or bag. This type of waste include human and animal anatomical waste, solid waste, examples body parts, tissues, surgical waste. This treatment method include incinerated or subjected to deep burial in areas where incineration is not feasible. Incineration is preferred to completely destroy infectious material and prevent environmental conditions. The purpose is to safely manage highly infectious and pathological waste that poses a significant infection risk. This red bin or bag, this type of waste contain contaminated recyclable plastics. Example catheters, intravenous IV sets, urine bags, oxygen tubes, syringe without needles and other disposable medical device made of plastics. These plastics are usually sterilized by autoclaving, microwaving, or chemical disinfections before being shredded and sent for recycling or further disposal. Blue bag or blue-white bag include sharps and glasswares, needles, scalpels, blades. These sharps are collected in puncture-proof containers and sterile using autoclaves or microwaves. After sterilization, they are shredded or encapsulated before final disposal. Glassware is also sterilized and disposed of carefully to prevent injury. Black bean bag, these contain general non-infectious waste that is not recyclable or requires special treatment. Discarded medicines, expired or unused, incineration, ash, chemical waste, laboratory reagent, all this include in black bag. Chemical waste is treated chemically or sent to secured landfill site. Medicines are destroyed using chemical or incineration method based on their composition. This diagrammatic representation of waste segregation, these four colors I already explained in color coding system. This diagram summarizes the guidelines for segregating solid biomedical waste using color-coded beans, highlighting both types of waste and appropriate disposal method. Here is an example. Black bin contains non-infected cytotoxic drugs and chemical waste. This red bin plastic containers, it include infected solids, dressings, all this which is autoclaved or deep burial. Yellow bag, it is anatomical waste, placenta, pathological waste, body parts. This uses deep burial. Blue or white, blue bag contain infected plastic, syringe, gloves, plastic weights. Its purpose is prevent reuse of infectious plastic items and ensures final recycling after disinfection. White bean, this white bin contain plastic needles, plastic containers, and its purpose is to protect waste handlers from injuries and infections due to sharp objects. So, color coding is essential for immediate identification and proper handling of waste. Each bin is associated with distinct waste types and specific disposal protocols to maintain safety and hygiene. Procedures such as chemical treatment, autoclaving, burial, recycling, and common facility handling are used to minimize environmental impact and infection risks. So, this diagrammatic segregation helps hospital and medical facilities comply with biomedical waste management regulations. Collection and storage. The collection of biomedical waste involves the installation of different color-coded containers for biomedical waste obtained from varying sources. The containers or bins should be placed in a location so that 100% collection is achieved. The symbol in biomedical waste management is generally used as a warning to take precautions while exposing to those substances. The biohazard symbol was developed by the Dow Chemical Company in 1966 for their contaminant products. So, subsequent to collection, the biomedical waste is stored in specific containers and stored in a proper place. The extent of storage should not exceed beyond 24 hours in big hospitals, contains more than 250 bedded, and 24 hours in nursing homes. Each container must be clearly labeled with the location being mentioned in them. The purpose of labeling is to trace the waste at the source. Storage spot must be clear with a warning sign. Transportation. So the collected waste are transported in trolleys or in enclosed wheelbarrow for treatment. The operator should ensure to avoid manual loading. The bags containers biomedical waste must be tied before hauling for treatment. Vehicle used for transporting should be special to avoid contact and direct contact with the operators, scavengers, and the public. So while transporting the container, it must be properly enclosed. The effect of accident should be incorporated in the design. So we'll start with trolleys. The use of trolley will make to eliminate the infectious waste possible at the source itself instead of accumulation. A new category of waste, these wheelbarrows. Wheelbarrows are used to transfer the waste from the point source to the collection centers. There are two types of wheelbarrow, covered and open. Wheelbarrow, this prevent corrosion care should be taken. Liquid waste from spilling in the wheelbarrow. So wheelbarrow also comes in various sizes depending on the utility. And chutes and dustbin. It is very important to calculate the amount of waste generated at each point. Dustbin should be of such capacity so that it can be placed at the specific site and they do not overflow between each cycle of waste collection. Second is minimize manual handling and spillage risk. So the process should limit direct contact between workers and the waste. This means using tools, all these tools, trolleys, wheelbarrows, dustbins, two instead of hands whenever possible. It protects workers from accidental injury and infection. And it should also reduce spillage risk. Proper equipment, leak, sealed, and stable bins should be used to prevent the waste from spilling during transport. Treatment and disposable method. Treatment before its final disposal of biomedical waste, it must be disinfected. Anatomical waste can be disposed by deep burial, syringe to be cut and chemically disinfected with 1% bleaching powder solution at the source of generation before final disposal into shark pits. Infected plastics to be chemically disinfected or autoclaved, shredded and recycled and sell for final disposal into the municipal dumps. So this incineration, most of the hazardous biomedical waste treated by the method of incineration to reduce organic and combustible waste to inorganic combustible matter. Incineration is a high temperature dry oxidation process that results in significant reduction of waste volume and weight. So the advantages of incinerator includes high reduction of waste volume in addition to good disinfection competence. It helps to save the space in the landfill. The ash generated can be disposed of safely in the landfills. Second is autoclaving of biomedical waste. Autoclave treats the biomedical waste through the mechanism of disinfection. The biomedical waste was subjected to following temperature and pressure based on its residence time. Third is microwave treatment. This microwave treatment uses a frequency of wavelength of 2450 megahertz and 12.24 centimeter respectively for the destruction of microorganisms. The infections contaminants in water with biomedical waste are destroyed by heat conduction when it is rapidly heated by the microwaves. By bacteriological and biological tests, the efficiency of microwave disinfection was ensured regularly. This chemical disinfection refers to the process of using chemicals to kill or inactivate harmful microorganisms in biochemical waste before final disposal. So chemical disinfection include bleaching powder, calcium hypochlorite and reagents. Chemicals like bleaching powder, sodium hypochlorite or chlorine compounds are commonly used as disinfectants in hospitals. These work by releasing chlorine, which has strong oxidizing properties. The chlorine destroys cell wall and essential proteins in bacteria, viruses, and fungi, which make the waste safe to handle. It is important because, especially for treating liquid medical waste or contaminated materials that cannot be incinerated or autoclaved. That is why chemical disinfection is important. It minimizes the risk of spreading infections and it is effective against a broad spectrum of pathogens. Deep burial Deep burial process is done in pits about 2 meters deep. These pits are half filled with waste, 50 centimeters soil, and then with waste. The pits are covered with wire meshes. When waste are added to the pit, a layer of soil shall be added to enclose the waste. The deep burial site should be impermeable with no shallow well in the nearby area. So the pit should be away from the habitation to avoid infection to surface or groundwater. The site selected should not be a flooding or eroding zone and should be approved by the authority. Last is inertialization. Inertialization, assimilation of waste with cement and other substances before disposal is called this process. This decreases the risk of entry of toxic substances into surface or groundwater. A typical percentage of the mixture is 65% pharmaceutical waste, 15% cement, and 5% water. A homogeneous mass is created and tubes or pellets are produced and then stored. This process is economical and not suitable for infectious waste. Advantages and limitation: incineration, high volume reduction but costly and polluting. Autoclave is effective disinfection but no volume reduction. Microwave is eco-friendly but expensive. Deep burial method is low cost but it is of risk of contamination. So, summary: biomedical waste is hazardous if mismanaged. Biomedical waste can contain infectious materials, so if these are not handled correctly, they can spread disease, pollute water and soil, or cause injuries to healthcare workers and community. Proper classification, segregation, collection, and storage is essential. The first step in safe management are identifying or classifying types of waste. This classification, like infectious, chemicals, or sharp materials, and keeping different types segregate, proper collection and storage in appropriate containers so that risk of accidental exposure, injuries, and contamination will be avoided. Color coding system ensures safe handling. Color coded bins are used so anyone handling the waste knows what is inside and how to treat it. For example, red bins might be used for infected materials, black for chemicals, yellow for anatomical waste. This system makes segregation easy and prevent mistakes. Various treatment method, incineration, autoclave, microwave, etc. will be used. Choosing the right treatment ensures that both infectious agent and hazardous chemicals are neutralized. Aim is to minimize risk to humans and environment. The core goal is to protect people and nature. Properly manage biomedical waste lowers the risk of infections, injuries, toxic exposures, and environmental pollutions. 1 minute. Question and answers. So I have listed 10 questions for 3 marks. Define biomedical waste, mention its two major hazards. Here you can define biomedical waste explanation and mention its two major hazardous products. Second is list any three major sources of biomedical waste. Third, differentiate between hazardous and non-hazardous biomedical waste. You can differentiate and give each example for each. Give two examples each of infectious and pathological biomedical waste. Give examples of infectious and pathological waste and why it is important. State the importance of segregation in biomedical waste management. Write the color coding system for yellow and red bags with example why it is needed and how it is convenient to dispose in yellow and red bags. Include examples. Mention storage time limits for biomedical waste in hospitals and clinics. It should not exceed more than 24 hours so that odor and infection will increase. List any three transportation precautions for biomedical waste. Any three transportation which is necessary and precautious for biomedical waste you can include here. Give two advantages and one limitation of incineration. Last is name two chemical disinfectants used in biomedical waste management. 5 mark question. Explain the classification of biomedical waste with suitable examples. Second is describe the color coding system used in biomedical waste management. Third, discuss the steps involved in biomedical waste management from segregation to disposal. Fourth, explain in detail the method of treatment and disposal of biomedical waste. Write a note on advantages and limitations of incineration, autoclaving, and microwave treatment. In detail, you have to mention everything with examples. Ninth is explain collection and storage of biomedical waste with safety precautions. 10th, discuss the health and environmental risk of improper biomedical waste management. Last, write short notes on deep burial method and inertialization method. Thank you, sir.
Yeah, thank you, madam. Thank you so much. So I think most of the topics you have covered here nicely.
Yes, sir. I try
Yeah,
to cover all biomedical
yeah.
waste.
And one more thing today, what we have seen is, and one more thing, your qualification you mentioned as PhD microbiology.
Yes,
Actually,
sir.
I was
Correct.
thinking that MSc MLT, but it is
No.
nice to see that you have done my
Yes,
PhD
sir.
microbiology. So I
Yes,
can
sir.
call you as a Dr. Namira. Okay.
Sir, my thesis is still pending, so
No issues, but because already it is
yeah.
in process, so then
Yes, sir.
I can call you as a Dr. Namir.
Yes, sir.
So
Thank you.
today you bought very nice presentation that is AIMS Delhi, madam.
Yes, sir.
They are segregating 74,500
Yes, sir.
kilograms of waste per month.
Per month,
So in that too in 2018. So I
18,
think
correct.
most probably this year, I think they have,
Increased,
it
yes,
might
sir.
have increased, yeah, it might have decreased
Mm-hmm.
because
Mm-hmm.
once they start segregating properly, no, so
Mm-hmm.
then we can reduce the quantity also,
Reduce the
I think
quantity.
so.
Correct, correct.
So coming to our students, we
Yes,
need
sir.
to recommend them to
Yes,
go
sir.
through the guidelines which were issued by the Biomedical Waste Management.
Correct,
That
sir.
is the Pollution Control Board. Guidelines are
Pollution.
there.
Mm-hmm.
2016 also one guideline is there.
Guideline is
And
there.
after that they have updated regularly.
Mm-hmm.
And
Regularly.
coming to segregation, collection, storage,
Yes.
transportation, treatment, and disposal.
Yes,
So
sir.
these areas, I think they need to focus very
Important
as
topics.
important
Yes, sir.
thing. And
Yes.
after that, coming to one more thing.
Yes, sir.
Now one new thing has come up, madam. That is green hospital. We call it as a green hospital.
Green Hospital.
So
Yes, sir.
then, what is the main concept here is, no?
Mm-hmm.
So to reduce the waste which was generated by the hospitals.
Mm-hmm.
So in the form of reducing the usage of plastic,
Plastics.
yeah, reduce the usage of electricity, yeah, alternative mode we can use,
Mm-hmm.
like
Yes.
solar lighting system we
Mm.
can use.
Yes.
And coming to some, because all these things are to reduce carbon footprint. That's the main thing actually.
Mhm.
So very nice presentation, madam. Thank you so much.
Yes, sir.
Uh,
Thank you.
okay, madam. Shall we wind up, madam?
Yes, sir.
Yeah, madam. Thank you.
Okay.
Thank you so much, madam.
Thank you. Thank you, sir.